Hello again, everyone, and welcome back to our final Q&A session. As Brett mentioned, we are delighted and honored today to be joined by Dr. Jennifer Doudna. And I'd like to give you a quick snapshot, and it's a snapshot of a much longer CV, but Dr. Doudna is the Li Ka-shing Chancellor's Chair and a professor in the departments of chemistry and of molecular and cell biology at UC Berkeley. Her co-discovery of CRISPR-Cas9 genetic engineering technology has revolutionized human and agricultural genomics research forever. And we are honored. We are honored to have you here with us today, Jennifer. So just to get our bearings and a little bit of the background, I know that in all of our lives, there are those serendipitous moments that we look back on and say, wow, if that didn't happen and that didn't happen and that didn't happen, I wouldn't be here now. I wouldn't be the co-discoverer of CRISPR-Cas9. And I'd like to know, and this can be from decisions your parents made, you made as a child or as an adult, education. What are those serendipitous moments that you reflect on from time to time?
Well, first of all, hi, Kathy. It's a real pleasure to be here. And I think, the serendipitous moments are really what make life interesting. And, maybe some of our listeners have seen Back to the Future with the, the scene in there about, taking a trip back in time and changing one little thing that just changes everything in the future. And I think, it is fascinating to imagine how my experience, would be different if little things had happened differently. And I guess when I imagine that, I have to say that one of the things that really made me who I am is my upbringing in Hawaii. So I grew up in a small town, rural working class town, Hilo, Hawaii. It's not one of the fancy tourist destinations, but it's an extraordinary place with amazing people. I had the good fortune to encounter some incredible teachers in my public school education there who encouraged my interest in science. And of course, my parents, especially my father, I think, was somebody who really loved learning, and he was an educator himself, not a scientist, but loved to think about and read about science. And so with his encouragement, I ended up, pursuing my dream of becoming a biochemist. I didn't even really know quite what that was. It just sounded cool. And, I really wanted to understand the fundamental chemistry of living systems in some capacity. And, I think when I think about what happened, all the things that happened after that eventually led me to CRISPR, I have to say that I truly met extraordinary people on my path who were incredibly passionate about what they did. And I kind of shared that with them. So, from my college biochemistry professor to my PhD advisor, and then eventually to my meeting with Emmanuel Charpentier, which, was in some ways kind of a serendipitous event, but led to our collaboration to work on CRISPR together.
Yeah, that's exciting. Now, at ARC, we've been studying CRISPR, gene editing, gene therapies generally, for a while now. But I'd like to hear from you how you would describe CRISPR-Cas9 for those who may not have that great knowledge of it.
The way I like to describe CRISPR is as a tool for editing the code of life. And what does that actually mean? Well, it's basically a way to make very targeted changes to the DNA in cells that allow scientists to manipulate genes, not only one at a time, but multiple genes at once, and do things ranging from changing a single letter in the code of the DNA of a human cell to altering 20 or 30 genes in a plant cell that allow the plant to grow under drought conditions and be resistant to pests. So those are just a couple of broad-ranging examples of what can be done with CRISPR. And it all came from a bacterial immune system, which during the current pandemic seems particularly fitting.
Absolutely. Was there at any time a Eureka moment, or were you on the hunt and you knew you were onto something big? When did you understand this was transformative, and what was that moment?
I guess what I've always loved about science is that you don't know exactly where things are going, or at least I never seem to. And, I always try to pick projects and problems that I find interesting and that I think relate in some way to very fundamental questions about biology, such as, why are we here? And, how did we get here? How did life get going in the first place? And why does it look the way it does? And CRISPR is no different in the sense that I started working on it back in, I'm sorry, get a little feedback there. Can you hear me?
Yes.
Yeah. Okay. I started working on it back in the mid-2000s when just a few scientists around the world had noticed evidence of a possible bacterial immune system in microbes that seemed very interesting and to me seemed likely to shed some light on the way that organisms evolve to avoid being destroyed by viruses. And so we started to work on this, and then fast forwarding a few years after my meeting with Emmanuel Charpentier, it was I still remember that meeting. It was one of these extraordinary moments in your life when you have a feeling about someone and about something like a project that is almost like a premonition of what's to come, because we met at a meeting in Puerto Rico, and we walked around old San Juan, Puerto Rico, those lovely cobblestone streets after lunch, a lunch meeting that we had, and it was just the two of us. And we started talking about the possibility of working together to understand the molecular function of a particular protein called Cas9 that is part of this bacterial immune system known as CRISPR. And in that conversation, which I still remember, it was one of those times when I just had this sense that there was something really, really interesting there. And so, we started to work on it together. And within not very long after starting that collaboration, it was quite clear to us that this project, which started as a very fundamental scientific collaboration to understand something quite specific about the biology of this immune system, was going in a very different direction, because once we understood how this Cas9 protein functions, it was clear that it could be harnessed for a very different purpose, namely for triggering targeted changes in the DNA of cells.
And did you, and because of that, I mean, you're a scientist, so the obvious conclusion was, wow, we could cure disease, we could change the course of life here for someone? Wow.
Yeah. No, it was, really, I think just there kind of was a moment, really, where with my coworkers in the laboratory, we saw the data that we were collecting for the function of this protein and looked at each other and said, this is just extraordinary. This is a protein that can be told where to go in a DNA sequence and programmed, and that can be controlled by the scientist now because we understand how it works.
It's incredible. So as we are embarking on this adventure, and I say that from an investor from a financial point of view, because it has been an adventure to learn about this and to invest in it. It's really interesting how, I'll go out there saying, hey, this has happened. And it's just like, it's eyes glaze over, or disbelief, just disbelief, because it's never happened before. So that's what's so interesting. But we are, so there's there are different flavors of CRISPR that, as an investor, I'm learning. We've got Cas9, we've got X and 10 and 12 and 13, and there are lots of numbers. Can you explain the differences here? And it seems like there's been some stabilization in the short term around Cas9. That's where most of the work has been done. Can you describe the differences and where you see this coming? Is Cas9 fleeting in terms of its benefits, or will it be used for certain things and then others for other diseases?
Well, as you mentioned, Kathy, there are a number of different proteins in these CRISPR immune systems in bacteria that all have a, at least at a high level, a pretty similar ability to recognize a sequence of DNA and trigger a change in that sequence in cells. And, there are different numbers associated with them, but Cas9, was the first of those to be investigated. And, again, back to your very first question about serendipity, this is one of those serendipities, that Emmanuelle Charpentier is a medical microbiologist, and she was studying a particular type of bacteria that infect humans. It turned out that bacterium uses a version of the Cas9 protein, the first, protein that we started to investigate together, that even today remains the best for genome editing. Who would have thunk it, right? It's just one of those, serendipitous things. Shouldn't have been true, but is. And so right now, what's happening is that what I see in the field is that there's obviously been lots of interest in expanding the toolbox, which is what a lot of these additional Cas proteins do. They provide additional ways that genomes can be manipulated, and frankly, depending on the particular use of the tool, some of these can be great. I mean, they really help to expand the kinds of things that one can do with the CRISPR technology. But that being said, in general, when the average lab reaches for a genome editor to do something, in general, they reach for Cas9 because it does the job very well.
That's great. So in terms of the potential for CRISPR to change medicine, we've done a lot of work on monogenic diseases, and are wondering if you think that CRISPR could be approved as a therapeutic intervention to rid us of disease, to actually cure disease. And if so, which kinds of diseases, or are we talking about all kinds of diseases?
Well, the short answer to your question is yes. I absolutely think it will be approved as a way to correct disease-causing mutations and in principle even to cure genetic diseases that in the past were untreatable and certainly not curable. This is truly extraordinary. I mean, it's just an incredible moment in biomedical history, and really the history of us as humans, that we now have the ability to rewrite our own genetic code. I mean, it's just quite remarkable to think about. And then think about what diseases are likely to be the first to be treated in that way. It'll almost certainly be diseases that are where there's a very well-known single gene that causes the disorder, and frankly, where the cell type that is affected in that disease is something that's pretty easy to get at. Mm-hmm. And so one of the categories of diseases that sort of check both of those boxes are diseases of the blood or the immune system, where one can imagine using a gene editing tool to correct a disease-causing mutation in cells that could be taken from a patient, edited, and then replaced, so that you avoid the whole issue of having to deliver into the body and make sure that the delivery is efficient, et cetera. That's something that is still very much on the kind of the bleeding edge of the technology, I would say. So I think you probably maybe a number of the listeners here heard about treatments for sickle cell disease that are going to be based on genome editing. And in fact, there's a woman named Victoria Gray who has been in the news lately, whose sickle cell disease has apparently been cured by genome editing. It's quite extraordinary. And she seems to be in good health, and so far so good. And it's been a number of months that she's since she received that treatment. And the thing to appreciate is that this is truly a cure, in the sense that traditional therapeutics require chronic delivery. Somebody has to take a pill every day, or they have to go and get an injection every month or something like that. And the idea with genome editing is fundamentally different, in the sense that this is a one and done kind of treatment, where the corrective mutation is introduced into the cells where it's needed, and then the editor goes away and the cells are happy, and the person is then disease-free. So it's really a change, not only in the technology and kind of an advance, really fundamentally, in the way we think about genetic diseases, but it's also, I think, an extraordinary time when we're going to see a real change in healthcare and in pharmaceutical companies and insurance companies that will have to think about, how do you price a technology like that? How do you price a treatment that isn't going to be delivered over the course of somebody's life or many months, but is in fact a one-shot thing? Nonetheless, it has an extraordinary impact on their life, just like a chronic treatment would be. So anyway, I think that's one of the many, very interesting aspects of this technology that remains to be figured out.
So we are studying the convergence of sequencing and artificial intelligence and CRISPR gene editing. We're seeing all kinds of platforms converge here, but this is probably one of the most important set of convergences. Is it wild-eyed to be thinking that as this convergence evolves and we get deeper and deeper into understanding what's going on, that we could cure most diseases? Is that outrageous to think? Or I'm just dreaming, I realize it's not 50, it's 15, 20, maybe a long time away, but is it a crazy thought?
Well, I would, it's not a crazy thought, but I would modify it a little bit, just to be clear. So I think that if we start imagining what happens in the future, in the coming years and decades, I think it's not unreasonable to imagine that genome editing will allow diseases that are caused by a single gene to be corrected and cured. So that is just, it's kind of mind-blowing to think about it. And it really does allow a very clear path to be envisioned, at least, for personal medicine, because this is really about fine-tuning. You can take this tool and you can literally program it to make the targeted change that one wants. And so that can be tailored to individuals based on their DNA sequence. So that's where the DNA sequencing obviously comes in.
Right.
But that being said, of course, there are many diseases and conditions that result from multiple genes that are the way they are, and then of course lots of diseases like COVID-19 comes to mind, that are not caused by genetics necessarily, but are caused by some external infectious agent. Although it's possible that that one could use genome editing there to help boost up the immune system of affected individuals. So, I think that there's really lots of opportunities, but my guess is that initially the targets for genome editing in biomedicine will be focused on monogenic diseases, because those are the ones where it's easiest to envision how this technology will address those.
Okay, and before we leave that, we are going to get a number of milestones from different companies this year. There are lots of tests out there, human tests, in the United States, which is very exciting. And from my point of view as an investor, it's no news has been good news from some of these. That's just I might be wrong on that, but how should we interpret the data coming out of these trials, and what endpoints? I know you're very focused on safety first and foremost, which you've commented on, but what endpoints are you really eager to see?
I'm just, I'm thrilled with how things are going so far. As you say, it's early days, but it's great to see that so far these genome editing applications in humans have been safe. That's absolutely key. And we've seen that now from several trial announcements. It's not just one. And so, and different indications, including for cancer patients with, T-cell therapy. So I think that, that certainly bodes well, and, again, we're talking about small numbers of patients so far, but as you said, Kathy, no news is good news, right? So that's certainly encouraging. And then I think what's coming down the pike is that, as you also said, we're going to start to see announcements about larger numbers of patients, and as these trials move ahead into the actual efficacy stage of phase 2, that we'll start to really get a sense of how well these therapies are working, and we'll see what happens. I think that right now there's lots of excitement in the field, lots of anticipation about the outcomes of these trials, and my expectation is that, assuming that we continue to see progress as we have, that those announcements, if positive, will really, encourage a lot of other people to jump on the bandwagon and get involved, because they will understand that, the opportunities right now are just expanding so quickly.
Yeah. And you mentioned COVID-19. So moving on to, anti-infectious agents and so forth, and diagnostics. How has COVID-19 changed your life and the life of your, researchers, and how has your focus changed because of COVID-19? Is there something you can do? I know you've been working on this to really help alleviate the situation for the long run.
Well, I'm a faculty member at the University of California, Berkeley, and one of the hats I wear there is I run an institute called the Innovative Genomics Institute that is a partnership between UC Berkeley, which is a research university, and UCSF, which is obviously fantastic medical school. And when the pandemic was coming into focus here in the US, in the early part of this year, we sat down at the Innovative Genomics Institute and asked ourselves, what could we be doing as scientists to address this global health emergency? And the very first thing that I think everybody got on board with was, we need to be doing a lot more testing. And so even though it's not directly related to genome editing, we thought, look, we need to set up a testing lab and be able to run clinical tests to let people know if they're infected with this virus. And so that happened in an extraordinarily short amount of time. This would be, you have to appreciate, we did this at UC Berkeley, which does not have a medical school. And so to do that and set up a clinically certified lab with all of the regulatory approvals at the local, state, and federal levels that are necessary was no small feat. So I have to really, my hat goes off to my team. They're just incredible, the people that got this done. And now we are running literally, thousands of tests around the Bay Area. We work with healthcare partners that serve primarily the underserved. We're very pleased to be able to do that. So we offer free COVID-19 testing for people that are in homeless shelters or encampments, people that are uninsured, folks that are first responders that are out, keeping our power plants running around California, for example. So, these are all people that I think really have benefited from the regular type of, testing that we can offer to them. And in fact, at the same time that we were doing that, we realized that to be ultimately really impactful on opening back up the California economy and including our local campus environment, we really need to transition that test to a simpler sampling method. And so what we're doing now is saliva-based testing. So you may know that that's not the typical way that people get tested for COVID-19. They have to go and get a nasal swab or sometimes an oral swab, and they have to do it at a doctor's office. We have now instantiated a saliva-based test that we're running as an experimental study. And our first test collection kiosk opens on the Berkeley campus tomorrow.
Oh.
So we'll see how that goes. We have a whole team that's been doing this, and the timing is good because it coincides with the announcement from the UC Berkeley chancellor that our campus is reopening for research, and in the fall we'll be opening up at a reduced capacity for undergraduate education. So my hope is that we continue to advance the surveillance testing and providing that as a service. But on the research side, back to CRISPR, one of the things that, as I mentioned early in this conversation, CRISPR is an immune system. It's a way that bacteria find and destroy viruses. So we thought, gee, wouldn't it be great if you could use CRISPR as a way to tell us if there is a virus in a sample? And so this is a project that started years ago, actually, but has been now adopted by a number of companies and many academic labs that are working on CRISPR diagnostics. And so we have also a pretty large team at the Innovative Genomics Institute doing this work, and it includes scientists on both sides of the San Francisco Bay. We have people at the Gladstone Institute working on it, and people at Berkeley and UCSF. And then we've got a number of corporate partners as well. So my hope is that will lead to a point of care or potentially even an at-home test that would be very fast for detection of the virus that will allow people to test themselves and ensure public safety through just very frequent testing.
Oh, thank you. As an investor, my another crazy question: would it be crazy to think that we could eradicate viruses long term?
Well, if you let me speculate for a moment, one of the things that we talk about mostly when it's either late at night and you might be having a glass of something, or when you're sort of speculating and fantasizing about the future, is we think about the possibility of what I call genetic vaccination. Imagine that it were possible to program immune cells in an individual so that they were very effective at fighting off diseases that person might be otherwise susceptible to. And I don't think it's a totally crazy idea because, let's take COVID-19. There's already a lot of interesting evidence out there that certain types of people are more susceptible to this virus than others. And we don't really understand the reasons for that. There's a lot of speculation and not a lot of data quite yet. It's quite early still, but you could envision that some of that at least may be genetically based. Some of it may have to do with the way that some people's immune systems respond to the virus or even overreact to the virus. And so if that could be understood genetically, it's possible that that could be also controlled genetically with a tool like CRISPR. So I'm fascinated by that idea. And again, just to be clear, I'm not saying that's coming next year, but I do think that's the kind of thing that over the next 5, 10, 15, 20 years, we're going to see very steady progress in that direction.
That's great. And then just quickly back to cancer. You mentioned, I know that there are a lot of blood cancers. Do you think CRISPR could apply to potentially, I know long-term, I want to be careful in saying that, to solid tumors as well? Is there any reason it couldn't be?
I think that's a very exciting direction in the future for CRISPR, I have to say, because as you alluded to, one of the challenges in the field of of cell therapies for cancer, so the cancer immunotherapy, is exactly this challenge of how do we deal with solid tumors. They typically don't have the kind of exposure to the immune system that would allow a robust immune response to develop, although there are very interesting sporadic cases of people with solid tumors where they do have a spontaneous, recovery. And that's been pinned on, a chance thing that happens with their immune system that allows them to fight that and destroy that tumor. So we know it's in principle possible. It's just a question of how do we make it possible for everybody to have that kind of a reaction to cancer. And so I'm quite excited about this. I think that there's still a lot of fundamental research to be done to understand the types of changes that would be needed in immune cells to achieve that kind of an outcome with solid tumors. But having a tool like CRISPR is certainly an enabling step in that direction. And I think that's something that we'll see real progress in the coming years.
And then on to Germline. I know that in your book, and so plug for your book here, A Crack in Creation, Gene Editing, and the Unthinkable Power to Control Evolution. I don't think it needs a plug, but you did say that someone said, someday we may consider it unethical, and I know ethical is a big consideration for you, may consider it unethical not to use germline editing to alleviate human suffering. So just wanted to know what you think about germline editing now and its potential versus the ethical considerations. And I'll just add here, many times I get questions as an investor about this topic, and some people want to rule out investing in the gene editing companies because of the ethical considerations. And we point out how strongly the focus is on that, in order to allow the science to continue to move forward. So any thoughts on that?
Yeah, that is a very critical point, I think, that you just made, which is basically that I think that, the vast majority of people working in this field or with this technology are 100% committed to seeing it used in a responsible fashion and ensuring that part of the motivation for that, of course, is as you said, to protect the ability of the technology to have impacts in people's lives that will be beneficial. That being said, anytime there's a new technology and it has extraordinary capabilities, as CRISPR does, there are some people that think that it would be interesting to do anything that's possible with that technology. And that includes, in the case of genome editing, making changes to the germline, human germline, which means alterations that affect the DNA in an entire individual. So rather than just, treating blood cells in someone affected with sickle cell disease, germline editing is fundamentally different in the sense that it means changing DNA in sperm or eggs or embryos such that the resulting person, if they were to be, actually born from those cells, would have those changes in every cell and would be able to pass them on to their kids and their future generations. So that's the difference with germline editing. And as you said, the companies that I'm aware of that are working in genome editing, whether they're in, essentially any field, any aspect of using genome editing in humans, are universally opposed to this. They don't want to focus on that type of editing, and in fact, they want to ensure that that type of editing doesn't happen right now because it just will distract from the real uses where it's going to have an impact in the immediate future in genetic disease. But again, there's always the question of, well, what happens in the future? And I think right now there's lots of things have happened in the field with respect to discussions around human germline editing, CRISPR babies that were actually born in China just over a year ago that had received CRISPR-induced changes to their DNA with the stated purpose of protecting them from future HIV infection, and all of the sort of concerns around that. And I think that what I see right now is that I think that one of the outcomes of that whole CRISPR baby debacle, frankly, is that it was really a wake-up call to the global scientific community that we can't ignore this. We can't not, if you're working in this area, you cannot sort of not be involved, right? It really requires the global community to step up and say, this is not okay. It's not the time to be using it in that fashion. And but I think also importantly, not to bury our heads in the sand about it, but to say, look, this is actually a potential application of genome editing that in the future, if it were shown to be safe, and if we knew enough about human genetics, and those are two big ifs, but if we did, you could imagine that it could become a way to prevent genetic disease in an individual and do so in a way that would remove a disease-causing mutation from a whole family. So for families that are affected by things like Huntington's disease, cystic fibrosis, there's a number of other obviously examples of diseases that are caused by a single gene that runs in families. Imagine being able to offer people a safe way to take that out of their their genealogy. It might become quite attractive, but I think that's clearly, to me, it's still quite a few years in the future.
Right. Yes. And now on to the business landscape, intellectual property. Having invested in disruptive innovation pretty much all my life, I've found two models here. Technology companies tend to be big on cross-licensing. There's a lot to do. Let's get on with it. And healthcare companies, it's a different culture, I would say. Do you think that we're moving into a kind of a cross-licensing world, or are just a few companies going to have the patents and will enjoy the royalties? And is the reason you founded 5 companies associated with this at all?
The patent landscape around CRISPR is complex, shall we say, and probably won't become non-complex for quite a while. That being said, again, that's no different from lots of industries where there's complexity around technology. So the good news is that has not stopped companies from getting founded and from moving ahead with the science that they feel is compelling for particular applications. My reason for founding 5 companies is actually that there's so many different uses of CRISPR that you couldn't imagine one company doing them all, or even 5 companies doing them all, right? It's probably going to take 500 or more. But I think that my excitement about the technology really extends beyond biomedical applications to agriculture and to what we call synthetic biology, being able to manipulate biological systems to do things like make green chemicals and make industrial products. So those are all uses of CRISPR that I think are kind of help indicate how broad based a technology this really is. I think that with regard to getting access to the intellectual property that all of the companies in the space are going to be utilizing, investors have had different approaches, as you probably know. Some investors are very conservative, and they say, unless a company has access, has licensed the foundational intellectual property, however you want to define that, we're not going to invest. Other investors have said, we'll figure it out later. It's too important. This is too important an area. We need to just get going, and it'll take years to sort all that out. And by that point, we'll have products. And once we do, we're going to go back and figure out who has the appropriate licenses and get FTO that way. So I think both approaches are, they are what they are, right? And I think from a perspective of a scientist, certainly, I'm just delighted that the science is moving forward the way it is, because I really want to see this technology used to solve real problems.
So thank you for that. I actually love that answer. And you mentioned agricultural technology. This actually, or agriculture period, this is a personal interest as well, if I understand about you farm tomatoes. Am I right on that?
You are right about that.
Yeah. So what what kind of, besides, I understand when you say being able to grow with less water, in drought conditions and so forth. One of the bad experiences investors have had are with GMOs and, genetically modified organisms. And, the Monsanto, now Bayer kind of lawsuits and everything. Can you describe why that is not going to happen here?
Well, I'm not sure it's not going to happen here, I guess is how what I would first say.
Okay.
I certainly hope it won't happen here. And that's certainly a motivation of my own, is to provide enough, we hope, information through, this sort of venue and others like the book and everything, that folks can make decisions based on actual facts and data rather than hype and, what's sort of, Frankenfoods and that sort of thing. But if it does happen, I think it would be a real shame, because what CRISPR does, in a nutshell, is it provides a targeted way to alter the DNA in plants that's different from how plant breeders work. And by the way, as I'm sure you and maybe a number of listeners here are already aware, all of our food is genetically modified, right? Yeah. Tomatoes are a great example. If you go look at what tomatoes looked like before humans started messing around with their DNA, well, they didn't look anything like what we have today. And so thank goodness there are plant breeders out there that, did start selecting for traits that make them big, round, red, and juicy. And so imagine having the ability to make targeted changes in plants that don't introduce a lot of other mutations along the way, which is how current plant breeding works, and could happen a lot faster. I think, to me, that's a good thing, and I think that there needs to just be a clear understanding of what this technology is really doing and what it's not doing. But that being said, it's a big challenge. And, you probably know that today, even in different countries, different parts of the world, GMO is defined differently. Right. And whether or not CRISPR leads to GMO plants is also defined differently. Mm-hmm. So same technology, but different definitions.
It's, yes, a challenge to be sure. So very interested. But, as you say about, or as you said that somebody said in your book, it would be unethical not to pursue this path.
Right.
Yeah, the same could be true here. I have just one more question. And before I ask it, I want to thank our genomics analysts, Ali Ehrman and Simon Burnett, for helping me formulate these questions. Couldn't have done it without them. So the last one, if you were an analyst in the healthcare space in the 90s, 2000s, 80s, 90s, 2000s, so recombinant DNA and all of that, what would you need to unlearn now in terms of understanding where healthcare is going, research, medicine is going? And what would you recommend really embracing now in terms of, new learnings?
Well, I guess one thought is it's going faster than you think. Mm-hmm. Change is coming faster than you think it will. That's really true. I can't tell you how amazed I have been with, and again, CRISPR is a great example of this, but it's certainly not the only example. CRISPR, the first publication about how CRISPR could work and work as a genome editing technology was in the summer of 2012. That's when Emmanuel and I published our paper. Mm-hmm. And now, fast forwarding, we're just about 8 years from that point. We've already seen people in clinical trials coming out of those trials with apparent cures to their genetic disease. I mean, this is extraordinary. And just think about the pace of that, how fast that's gone. And it continues to accelerate. You know, we've seen literally tens of thousands of publications in the scientific literature since that original work, that is all, you know, papers that are utilizing CRISPR for just almost anything you can imagine doing with a genome editing tool. And again, that continues to accelerate. It's just amazing how fast it's going. So that's one thing that I would certainly say. Another is that I think increasingly what I see is that different technologies are integrated together. So in the past, going and thinking back, you know, it really was the case, and this is very interesting because it actually was reflected in the way I used to run my research lab as well. You know, people were quite siloed. You know, they were working on different projects, and each student or postdoc had their own thing they were doing. No longer, right? Now, everybody in my lab, almost without exception, is working on a team of people. And why is that? Well, it's because instead of, you know, one sort of approach being taken to a problem, we're taking 5 different approaches and whooshing them together. And as a result, the project goes faster, it goes better, and we can do things that we couldn't have done in the past, or we wouldn't be able to do without all of those different work streams that come together. So I think that, and I'm not alone, I mean, I think that's sort of emblematic of the way a lot of research programs are running now, whether it's in academia or in companies. And so I would encourage investors really to pay attention to those opportunities to integrate different technologies. A big one that's coming down the pike, it hasn't quite gelled yet, but I think it will, is AI, you know, sort of machine learning and genetics, right? I mean, that's, you know, that's it's coming for sure. There's lots of excitement there. There's some hype for sure, but I think some of it's not hype. Some of it is really going to be quite transformative. So I would definitely pay attention to that.
Yeah, what you're saying is something that I am saying in the financial world too, that research departments are going to have to reorganize in order to understand and capitalize on these amazing exponential growth opportunities. And we have organized that way. We have. So Simon and Ali work with James Wang, our AI analyst. I mean, they're talking the same language. They're just coming at the world from different angles. So it's really fun, and I think really important. Well, Jennifer, you and Emmanuel are changing the world. So thank you so much for giving us this time, because the work you're doing is so important. And I know that's where you would, by and large, prefer to be. So we do thank you, because I think our world needs to understand more, because we want to allocate capital to its highest and best use. And there couldn't be a better one than this one. So thank you so much, and thank you for your contribution to this world.
Thanks so much for inviting me today, Kathy. It was great to talk to you.
Okay, great. Thank you so much. Okay, and with that, I'll hand it back to Tom Stout for our closing remarks.
Wow, wasn't that fantastic? I hope you enjoyed that as much as I did. That's going to conclude ARK Invest Big Idea Summit 2020 Volume II video conference for today. A big thank you again to Kathy Wood, Brett Winton, Jackie Reeses, and Jennifer Doudna for providing their time and expertise. And a big thank you to ARK's fantastic marketing team for putting this together and adapting to our new digital reality. Most importantly, thank you to you for joining us today. We hope the summit provided you with trusted insights from ARK's research ecosystem during these uncertain times. And as always, we encourage you to provide your feedback to us at info@ark-invest.com. As a quick reminder, a recording of this video will be made available on ARK's YouTube page. And if you want more disruptive innovation insights, we invite you to visit www.ark-invest.com, where we post all of our original research on disruptive innovation, or check us out on Twitter @ARKInvest. Stay safe and well, and on behalf of everybody at ARK, we look forward to seeing you next time. Have a great rest of your day.